been introduced to a lot of work that's happening in N-of-1 therapies with ASOs, et cetera. And we have a pretty extraordinary panel led by our friend and colleague and RareX board member, Walt Kowtoniuk. If you guys can just come on up, that would be great. Thank you. So we're going to let Walt, you're going to take it over. You guys are going to do your introductions. Thank you all for joining, and we'll let you take it away.
Thank you, Nicole. I'm going to ad-lib here for a second. Is Julia still here? Julia and I have a similar beginning of our journey to RareX. It all starts with Nicole. There might have been a drink involved, lots of hands flailing. She's going to regret she gave me a microphone after this. But a few years ago, Nicole approached me and asked if I wanted to help build RareX, and I honestly approached it with some skepticism. Nicole was waving her hands around, and turns out that was a pretty magical thing, though. And the thing that connected it for me, and it ties to exactly what we're going to talk about here, I spend my days trying to find ways to build medicines, to try to create therapies. Ultimately, it comes down to the financing of drug discovery. And so many times when we look at a rare disease and we think about ways to create medicines for rare diseases, we have this tremendous armamentarium now of therapeutic modalities. We've got CRISPRs and beams and ASOs and small molecules and antibodies, and you wake up tomorrow and David Liu will have another thing, right? And yet we're often stuck, okay, cool, but what do we do? What do we measure? What's going on with this patient? What's going on with this community? And I think that's really what got me to join this effort with Nicole and now Charlene and the whole RareX team is, if we can start systematizing and really enabling communities to, in a robust and specific and high-resolution way, define what's going on with their patients, it's going to lay the foundation and set the stage for drug discovery. And I think that's ultimately the end game for a lot of the people in this room and certainly a lot of the people on virtually. So I thank Nicole for having the vision to set this up years ago. I thank everyone for being here. I mean, this is tremendous to see the data that Charlene shared and how many communities and how many patients. This is nascent, but it's happening quick, and I think it's just a testament to the need and the opportunity here. And I think the last thing I'll share, I think in some ways, and I know I don't want this to sound bad, but in some ways, data is the easy part, right? I think it's so important that we are doing that, but what we're gonna talk about is how we start converting that data into knowledge, how we start converting that data into products. And I think that's really the next step for all of us, and it's really in the spirit of why we're all here today, the theme of the meeting being data, now what? So I work in venture capital. I spend most of my time thinking about how we're going to create products, how we're going to finance those products. Most of my time is spent in genetic disease. And one of the things that I'm really intrigued on as we think about the future of rare disease medicines is the long tail, right? I don't think I have to convince anyone in this room that rare diseases are rare. They're getting rarer and rarer as we identify them and segment diseases. We have to come up with new models, new ways of thinking about regulatory paradigms, new ways of thinking about commercial paradigms, new ways of thinking about paying for drugs. We're here today to talk about N-of-1 and specifically want to focus in on how patient-collected data, that patient data sets the stage for and enables therapeutic development, particularly in the N-of-1 space. So with that, I'm going to ask each panelist to introduce themselves, and I'm going to also ask them to say a few words about how, from their organization or from their perspective, patient data becomes so important for what they're going to do. And I'm going to start with Winston.
Sure. So I'm subbing in for Tim Yu today. He couldn't make it, but really is actually participating online and eagerly tuning in. We just heard from Julia this amazing story of Mila, right? And how Tim was the pioneer who took from end to end this, from diagnosis to creating a new molecule to go into somebody, and then design the clinical trial and dose Mila all within a single year, right? So that's an incredible feat. But what I want to emphasize of what's important about that is the pace was fast, yes, I agree. But it was the fact that it was a new medicine that was developed for a single person's genetic, their specific genetic mutation. So, we've heard about repurposing drugs. We've heard about taking assets off, shelved assets from biotechs. But this fundamentally was a new molecule that was developed for an individualized, for an individual. So I think this adds to the menu of what Walt was saying, that long tail, right? Because as RareX identifies, from 8,000 to 10,000, and then one day to 15,000, that's probably going to be added in that rare, ultra rare space at the long tail where you have just a few patients. And you can have the menu option of going and repurposing drugs and trying that, but you might want to seek out these N-of-1 trials with medicines that are specifically developed for that individual's genetic variant. So I think what I want to emphasize to you guys and set the stage for all of us up here is we're entering this era of programmable medicines where fundamentally you're using information molecules that may have a history that's tens of millions of dollars with years of development experience from biotech based on a commercially viable drug. But then we get to build upon all of that to actually reprogram it for Mila, for any of your loved ones. And that's a path we're just embarking upon today. So I hope that helps contextualize all of the work that Tim and so many others on this stage, from Sarah and nLorem to Chris at Creyon, are excited about.
Can you say just a few sentences on the N-of-1 Collaborative and what it is?
Yeah, so Julia gave a really compelling introduction, right? And the N-of-1 Collaborative is really based on this idea that, if we are going to go from Mila to millions and actually make individualized medicines routine, it can't just be the extraordinary circumstance of finding a Julia and finding a Tim and seeing them meet together on social media and then getting all the help from Ionis. It's like that's not a scalable and sustainable model. And instead, what we need to do is discretize it into these components of what needs to go into the process of making an individualized medicine, right? That means, Julie mentioned it really well, like how do you even select the patients? How do you do those preclinical studies? How do you make the drug, right? That's such an important part of it, and we shouldn't expect physician investigators to actually have to have that knowledge, especially when industry does it so well, right? And then designing the clinical trial, executing it. What if we could put together that playbook for anyone who is seeking that, that menu option of making an individualized medicine? If we can build that and be a resource for the community at large, that's what the N-of-1 Collaborative wants to be. Julia's point about the data is also a really important one because, like we said, it's insufficient just to have the, just say we're going to enable these trials without actually aggregating the learnings and being able to feed that back quickly in real time. We don't want to wait for a publication. We don't want to wait for biotech or biopharma to ungate that. So we're building a database and have a lot to learn from RareX, frankly, of how do you take all of that information, share it pre-competitively as well as in real time in that open science manner so that all of these trials, when they're being designed, your investigators can access them in real time. So,
thank you. Sarah, how about yourself and nLorem?
Sure. My name's Sarah Glass. I am Chief Operating Officer at the nLorem Foundation. I'm a geneticist by training, and I've been in rare disease drug discovery and development actually my entire career. In about 2020, I have a son who was diagnosed with a rare disease. So even though I had been part of the community, I became part of the community, and my son has an N-of-1 mutation. So all of these personal and professional passions, I think, all come together like many of you, I think like Wendy mentioned this morning. So nLorem Foundation is a nonprofit drug discovery and development organization that really focuses on and is committed to providing these drugs to patients for free for life. We focus on quality ASOs. We really leverage the experience. We were founded by Stan Crook, who pioneered RNA therapeutics like 35 years ago. Ultimately, in his retirement, decided that, antisense oligonucleotide technology, and like you've already heard, it's efficient, it's cost effective, it's versatile, it can be specific to mutations, and it's scalable. And so ultimately, what else do you want to do in your retirement, right? But start a new company. And I joined him last year because when I heard about this, and I think like many of you, you're like, wow, how can I help? How can I be a part of this? So what we're doing now is really industrializing this. We leverage I think we've talked about the experience, the 30 years of experience with ASOs for every single patient. So for us, when we talk about what the key components of data are for N-of-1s and for nLorem, I guess I sort of see it in 4 from the drug discovery perspective. The first is the fact that the reason we can even talk about N-of-1s is because we have an established chemistry, and the FDA has acknowledged that there is enough data to be able to actually draft 4 guidance documents to give us a roadmap to discover and develop and provide these drugs. So this is the only technology so far. I'm sure Winston will talk about, we'll have CRISPR and we'll have gene editing and others in the future. So that's the first piece of data. The second important data for us is really and truly around who can we actually help. And so in the N-of-1 space, it's about being very transparent. It's like we acknowledged ASOs cannot tackle every mutation. So what is the data that we need to be able to make that decision? The first thing is, do we actually understand the mutation? Do we understand the consequences of that mutation? Do we believe that that mutation, especially in the context of other mutations, is the pathogenic, the driving mutation, that if you target it, this will ultimately have clinical benefit? I think the other important component on really establishing from a drug discovery perspective, if we can help someone, is do we understand the trajectory of their disease, their clinical manifestations. Most of these individuals, to date, nLorem has accepted over 70 patients into our program, so that gives you a sense of really and truly the scaling that we're doing to be able to help many, many more patients. And most of the patients that we actually can't help that are applying to the program, it's partly because they may have a mutation, but we don't actually understand the consequence of that mutation. And so therefore, you don't understand if you actually target that mutation, if you don't know what the mutation does, how do you know if you target the mutation, what will happen if you're trying to treat it? So that's a really important point as well. I think the last thing I'll mention too, as far as data that's really important on the drug discovery side, and I know we'll talk more about the clinical research data, but it's just sequencing data. So we all, have diagnoses, you have sequencing, you may have targeted, genotyping, we have a whole slew of data. Most patients come in and say, all right, here's all the data I have. Fortunately or unfortunately, what is so important is the quality of all of the data that goes into drug discovery and development. And that's inclusive of the sequencing data that we actually need to use to be able to design the ASOs. So for every patient, depending on whether you're going to target the disease-causing allele or the whole gene, depending on whether the consequence of targeting the whole gene would still be acceptable for that patient, you need to have the whole sequencing data and then be able to design ASOs across that genetic material. We typically leverage that genetic data in a way to design upwards of 500 ASOs for every single patient. And I think really that's one of the core elements that we feel like is starting from before we even bring a patient's mutation to become a drug target. There's so much data that goes into it. And we're working very closely with, out of these 70 patients, I think this represents 30 different institutions, I think 50-something physicians. And so with every single physician and institution, a number of you are here today, really working on how are we ensuring that we're capturing this data. nLorem's model, because we are a nonprofit, we are not actually, the IND holder is the physician. We're not going for any market approval or commercial endpoints. Our goal is to be 100% transparent. And so Winston and I have had some really great chats too, just about how are we ensuring that as a community we're kind of merging this. It ultimately comes back to the patients deciding where they want their data to go. But ultimately, when the patients consent, the institutions and physicians will decide, and nLorem's creating a platform to be able to enable them. So.
Chris, can we come to you next?
No, sure, yeah. First, thanks for having me join the panel, and appreciate the conversation today. My background's in the interface of biology and computer science, and I'm the co-founder and CEO of Creyon Bio. Our goal is to really sort of address this long tail of rare disease. We appreciate that it's a crisis of need in terms of creating therapeutics, and we also recognize that we live in a world today where we have the promise of data. We can sequence individuals and understand the molecular and genetic basis of disease, sometimes within a day of a sequencing event. But what really drove us to create Creyon was this disconnect between the rate in which we can understand the molecular and genetic basis of disease and the time frames it takes us to actually turn that into a meaningful therapeutic, and do that in a way that is not so costly that it doesn't fit within the current healthcare system today. So what we're building at Creyon is a platform to engineer oligonucleotide-based medicines. That includes ASOs, siRNAs, aptamers, ultimately any sort of chemically synthesized polymeric nucleic acids, so that would include guide RNAs and the like. And the idea is to make it so efficient that it is, in fact, a commercially viable process where we can deal with all of the indications. And the promise, I would say, is that we shouldn't be thinking about N-of-1s. We should be thinking about the community at large. As we learned today, numbers matter. And pulling people out of the rare community because they happen to be in a disease population that only is afflicting 1 or 2 people, we'd like that not to matter. We'd like to make a platform that allows us to engineer compounds safe and effective at the highest bar of quality as possible, but not have to worry about whether it's for 1 person, 10 people, 1,000 people, or 1 million people. And so we're building out the data set to inform ML and AI models that allow us to then engineer these compounds so that we have really changed the priors going into the clinic. Right now, unfortunately, when you think about drug development, why does it take so long? Why is it so expensive? It's because we're actually, as a community, not very good at making molecules that we know are going to be safe in the clinic or can be effective in the clinic. And we go into a phase 1 trial, actually, and the sort of current industry standard is that you get up a 50-50 chance of it being safe and coming out of a phase 1 trial. How do we change that prior? And if we do change that prior, if we can go into the clinic and say, you know what, this compound has a 90% chance or a 99% chance of being safe, now you've really changed the risk-benefit conversation we're having with regulators. And that changes the way you think about advancing things from, I have an understanding on a molecular basis of the disease, I have a drug that I know will be safe and effective. Can I systematically start to erode the time and cost it takes to go from, I know what I need to do at a molecular level, at a genetic level, to I have a drug that I need to get into a patient. And now the conversation with the agencies and the regulators are not about, how do we assure what are the couple of different non-human species that we need to go through, or non-clinical data we have to go through. It's how do we get this there faster, because we've changed the risk profile. Now it's about risk versus benefit. If you know you have a drug that's safe, you can then say the risk of waiting is too much. Time matters. We need to get into these patients fast, and we can do that. And if we can do it safely, then we can have time on our side. And that's what we're aiming to build with Creyon, is a toolbox that allows us to turn a crank, create compounds that are ready to go into the clinic as fast as possible, so we have compounds in time to make a difference.
Luke, can you also introduce your organization and maybe even just jump to first discussion topic about how data can help bring people together?
Yeah, sure. Thanks so much. It's pretty incredible to be here with all of you and everybody out there. So thanks for including me, Nicole, and everybody else. I'm Luke Rosen, and I represent a very incredible community of people. When Susanna, my daughter, was diagnosed at the age of 2, we couldn't find anybody else with, certainly not with her mutation, and we still have only found a handful of people with her mutation. And it's a very large gene with many different mutations along the way, and each of them are different. I think the most difficult part of data, and this is something that I spoke briefly with Julia and some others, is interpreting what doesn't appear on the collection of data or the data set that happens. So we were very fortunate to be supported by this army of people when we were diagnosed, and our physician, Wendy Chung, and nLorem. We're fortunate enough to be part of nLorem, so I feel like the structure of my hands have changed because my fingers are constantly crossed. And so we are just incredibly fortunate to be part of nLorem and what Sarah and Amy and Stan and the team are doing in collaboration with Wendy. But, about a year ago, Wendy had our family start rigorously collecting data. And Susanna's disease is a neurodegenerative one, so it changes, and you can see that change in the data we're collecting. And we did it in a way that was very difficult. It was daily, and she has hundreds of seizures a day, and she falls a lot. So she's, I was talking to Charlene about, parent fails, and when you feel terrible about parent fails, like if some reason, we didn't bring her meds one day and something horrible happens, or if, she falls, right? And we're collecting these data, and it's so difficult every single day, how many seizures, the time points, the duration of the seizure, and then notes about the seizure. So we record ours by voice. I'm not going to endorse any brand, but it starts with, hey,
Alexa.
Susanna's having a seizure now, and we start describing it, and we have these, devices in every room and even in our yard. And so they're automatically time-stamped, right? So you know the duration of each event. But as we're counting, all of a sudden it goes from 100 to 110 seizures to 5 catastrophic falls that end up in the emergency room, and then to 0, right? And the 0 is because that's when we had to put her in her wheelchair for a week, right? And that's when she, started a new seizure med that she'll take tons of seizure meds, and now she's on 4 or 5, and she plateaus after 4 months, and then we have to go in and find a new one. And so that just happened to be the time that high-dose Valium at night took her out of her status overnight, and so she had a better day during the morning. So it's the continuity of data that we collect, and then when life changes, so does the data. And so if we're averaging numbers or we're trying to see trends in the numbers, all of a sudden when there's 0, it's not because she's gotten better. It's because she's gotten worse, and now she's in her wheelchair for the next, however many months. Or, if she's fallen and twisted her ankle so badly or broken a bone, she has more broken bones than I ever did, and I played hockey for 25 years, and she, it's 0 because she's, had a horrible fall. So I think incorporating those numbers that are seemingly exciting or seemingly better, but are really not, is something that doesn't happen, right? And if you're averaging and collecting and collating all of these data, how do you interpret a 0 as bad and not a 0 as good, is my biggest problem. But we are, like I said, just so fortunate to be part of this and to hopefully intervene with Susanna's mutation before her disease becomes beyond repair. And it is an army of people like Sarah and nLorem and Wendy and so many people in here who I don't even know, who are putting this epic amount of work into one person. And I think that there needs to be a way to scale this. And I think, if I could, I would turn it right around to Walt and say, how do you go about, as a VC, how do you go about looking at this that could be a successful model of treating people, and how do you fund it? Because that, at the end of the day, is how we end up getting there. Maybe a different conversation for another time, but so that's me, and I do reiterate that I represent a really wonderful community that has different mutations, different challenges, and not many of each. So, yeah.
I'll attempt to answer it, but then I'm going to ask Sarah to go. I think as I look at the opportunity around N-of-1 therapies as a commercial enterprise, investable business, I think there are a couple really big glaring issues. The first one is patient ascertainment. Unfortunately, we often find patients late in their disease after the symptoms have onset. And even then, there's often not enough genetic information to know whether nasal modality is the right way to approach that disease. And so we're constantly playing catch up, right? Which then ties to the second challenge, which is having confidence that if we do manage to intervene and this Herculean effort does come together, that we're gonna actually be able to measure benefit, right? If we ultimately want to create an investable enterprise, we're gonna have to measure benefit and make that claim to someone who will pay for it to pay for that benefit. Which gets to what I want to ask Sarah about here, which is as we think about patient-collected data and RareX and all of the great, rich information can be in there, how does a group like nLorem look at an application and think about this, whether this is a program or this is a case they want to take on, and how does the data that the RareX community and the communities working in RareX feed that assessment?
Yeah, well, it's a great question. I mean, our number one commitment is to focus on the technology. So that's our core expertise, is to say if we feel, and leveraging the nonprofit model, if we feel that our technology and our sort of capability in that space can target a patient's mutation, physicians actually apply on behalf of their patient. And so, we're not in the point right now, like you said, we're not actually deciding who comes to us. And there have been conversations even in some communities, a patient will come in and we have a very robust and rigorous assessment of the mutation itself, of all of the functional consequences, and then on the clinical status and a lot of engagement with that physician. So we need to understand specifically from that physician, how is this patient doing? What does their trajectory look like? Tell us about their clinical assessments over the last few years. Some of them, like Luke, come in and say, okay, well, here's our graphs and these beautiful, really devastating, tables and all these figures about data. But ultimately, I think we're really and truly guided by the technology and also the fact that I mentioned before the FDA guidance document. The guidance actually enables patient and caregiver reported outcomes. So, we've had one conversation with one physician recently. Their patient is 16, and they say, well, you know what the best assessment would be for this patient? His favorite thing to do is video games, and that's declining the most. It's fine motor skills. So how does one actually measure fine motor skills? In the N-of-1 space, really the goal here is to be able to determine what clinical outcome assessments should be measured, and our model is to collect it during the ASO discovery and development phase, and then collect the same assessment during the treatment to be able to compare. And really, because we're not actually going for a regulatory approval, we just need to enable the physician to make their decision on whether a patient is benefiting or not. What is that benefit-risk ratio? So I think, the key question, though, is if we're going to decide on any physician can and any patient can use anything that they think might be helpful, I'm sure Jason from across health, I know, and I think when we've talked all about these assessments, that sounds like a disaster. I'm sure, Nicole, you're like, oh. But, our goal really is to work together and collaborate. So really working with RareX, with Charlene and Nicole and the team, working with Combined Brain, with across healthcare to say, okay, let's try to say, even if video games, and this particular one is this particular patient's, key assessment the physician wants to use, can we at least try across patients with similar clinical manifestations to have, say, 80% common and 20% unique? So that's kind of the approach we're taking. Honestly, we're navigating the opportunity to be flexible and to be customized and personalized while trying to underpin that with the rigor that we think eventually, of course, we can't just do it however we think we want to do now and expect it not to change. So that's, a big part of this is like doing this and setting it up in a way that in 3 or 5 years, when we're collecting the data now, to say, okay, well, we have seizure counts on, how many, 50, 100 patients. What have we learned and how can we apply that to be able to say to insurers, hey, look, this is an approach that works, and you should pay for it, and Walt will fund it if you do.
Yeah, actually adding on.
Yeah, I'd love to know the N-of-1 collaborative perspective here, particularly thinking about
Outcome met.
the efficacy standards, the outcome standards.
I think the efficacy is such a key important, it's a key part of it, because you want to know when you're providing a treatment to a patient that it's doing something, right? At the end of the day, that's also the standard with which FDA evaluates its safety as well as the expectation of benefit. And so when you think about outcome measures for the N-of-1 on the N-of-1 space, right? I just want to share with you that from the clinician perspective, it's actually incredibly lonely developing an N-of-1 trial, right? You don't have that benefit of like, here's a 20,000 randomized controlled trial that's published in JAMA that I'm going to look at to figure out where my patient fits. It's the data from the community that really helps drive what are the important things. So I would say like 3 particular things, right? One is what's the baseline, right? Like tracking how that has either progressed. We know that even on the N-of-1 level, sometimes there's patients who are, atypical presentations, as Charlene said, within even a very small disease already. So what is your own baseline? The second is, what are the outcome measures that are meaningful to that individual and their family, right? For some folks, it's not about having fewer anti-epileptic drugs, but really, can we, communicate even through sign language? Like those are the meaningful measures. And then I would say the third thing of collecting these data is like, say comes the day when Chris shortens that timeline to ASO development to just say weeks, right? If you had collected all that data in the baseline beforehand with a line of sight towards what's meaningful in the clinical trial, that suddenly gives you time to apply the treatment earlier on and see that N-of-1 change rather than having to, spend time collecting all that. So anyways, my thoughts there.
Can I add something to that too?
Please.
It's such an important point, but the how we measure efficacy is so important too. It can't just be an arbitrary number, right? Because something incremental would change our lives in such a major way, right? And I think that, we very early on, our physician, who is just, Wendy, who's here, I think, she told us that, with this disease, if you look at it like a highway, there are going to be parts that fall off. And it's going to be, she's going to change. Susanna is going to be a degenerative course. And if you can focus on one lane and make that lane really strong, then we're doing the right thing. And I do this anytime I speak. I feel as if I'm a voice for my daughter, so I'm going to do this quickly. I was sitting with Susanna. She was on my lap, and we were in school, and she was watching her classmates who are neurotypical and don't get around differently the way she does in a wheelchair, do a dance recital. And I could tell that she was getting very self-aware. I mean, she's a kid. And I could tell she started moving her feet a little like she wanted to dance. And it was heartbreaking. And for her birthday, one of the first things I did is call Bina Shah, wherever she is, Bina. Crazily, there are not many ultra-rare families that we can find in the New York area, right? In the tri-state area, which is New York City. Susanna, one of the lanes that we focused on was speech, right? She's losing her vision right now, and things are very difficult. She's having a lot of seizures. She's falling. Things are getting worse and worse, but her speech is great. And this is a video of Susanna saying thank you for her dance class that she's getting with Bina and Karina. And we decided we were going to call literally the best dancer in the world. This incredible woman named Lauren, who is the Swan in Swan Lake, right? And so we got in touch with her, and she gave, Karina and Susanna a dance lesson. And here's Susanna's voice.
I'm really, really happy.
What are you happy for?
My dance class.
Yeah, we're so thankful for you, Lauren.
Yay! I'm so thankful for you to sign me up for a dance class.
So that's Susanna's voice. So things aren't always catastrophic and awful as it seemingly is. We have these beautiful moments of joy that we just hold on to, and that was one of them. So I just wanted to share that.
Thank you. Thank you. Yeah, hard to follow that. Before we go to questions, I just wanted to ask Chris one question, which is, as you think about what we're talking about here, think about what you and Creyon are trying to do, what do you think the next decade, what's the future of N-of-1 therapy look like in your mind?
Yeah, it's a great question. I'd like to say that the future should not call out N-of-1 therapies. It should be, one of the analogies I like to often use when thinking about oligonucleotide-based medicines, ASOs, or the like, is knee replacement or joint replacement surgeries. Every time someone gets a new knee put in, it's a custom therapy that gets installed in their leg, and it's not a Herculean effort. You go in, you get an MRI scan, they do, or a CT scan. It gets sent off to a manufacturer somewhere. They custom manufacture a joint for you. They install it, and you walk home, right? Or maybe you wheel out of the hospital and you walk in the next couple of days. The reason we can do that is because we understand the sort of the materials that go into those joint replacements, right? We know that the polymers, the metals, everything about it, we know they're safe and biocompatible. We also know how the surgery works. We know how to put it in. We know all the things we need to do to make that a safe and efficacious thing, something that we can do routinely over and over again, and we can do it in a way that is compatible with demonstrating benefit, and payers are more than willing to pay for it. We can do analogous things in genetic medicines in terms of sequencing. We can sequence a patient. We can know, it's not 100% yet, but often you get sequenced, you know what's wrong. And we're increasingly getting to a place where we understand the pathology good enough to understand that this modulation will have benefit. And where I think we're all thrilled is that oligos actually provide the tooling to actually make that change in the cell, to actually have traction to make a molecular modulation, a precision to make a drug that would be just as efficacious as a knee replacement, but at a molecular level inside of cells. But the problem right now is we don't know how to engineer them such that we have confidence that it'll be safe. So we have to go through this Herculean effort. So the future that I'd love to see is that we don't call out N-of-1s. These are just another collection of rare diseases that get pooled together. And the fact that you happen to have a disease that is caused by a mutation that we maybe saw once, maybe saw twice, maybe we find another 100 people. It shouldn't matter. We should be able to make safe and effective drugs that can be applied and help as soon as we know what to do at a molecular level.
Thank you.
I wonder if I could just add to that, because I
Please.
think I just want to sort of put an exclamation point on the safe and effective, right? Yes. So this is still drug discovery and development. This is a patient conference, right? We want to be very transparent, 100% open. Every mutation right now is its own drug. It's regulated by the FDA. We still know we need 13-week tox studies, things like that. So I love and I hope we can be here together in the next year, 3, 5 years and say, okay, look at this, we can do hundreds at a time. I think what that's going to take is a lot of data, right? It's going to be a lot of data. It's going to be Nicole and Charlene and all of us actually working together to say, here are the 10s and 20s and hundreds of ASOs we've discovered and developed. Here's the data, and here, FDA, here's how the process could be modified to be able to help more patients in the way that you're describing. But I think I just want to make sure that we're all being very clear, that we're all working in the same environment. The safety of these drugs is really important. And while we can say that, eventually this is a platform and we can just apply it quickly, we still want to be very mindful because it will only take a few missteps. And I think that's why we're all being very cautious, to be able to say, we're doing this the right way. We're doing this with quality. We're also aligning with the FDA. And I love your vision for that. So I just wanted to comment on that as well.
Great. And I think tying into the conversation we were just having about, how do you capture benefit? How do you integrate so that when we have these stories where we know that what the baseline is and there's a differential, where you see positive gradients, being able to sort of think about creatively, and I think where the RareX has a huge potential is to be able to allow us to sort of start to aggregate these success stories and maybe opportunities where it wasn't successful, so we can think about metrics and statistics that allow us to actually do clinical trials that are what we want, which is to demonstrate benefit that we're having on patients so that we can get payers to pay for drugs and we can work within the healthcare system we live with. And I think this is where the data collective that is being pulled together by organizations like the N-of-1 communities and RareX are critical.
Awesome.
Nicole and Charlene have mics, and I'll just say while they're going around, I'm full of hope with the four of you here. We have nLorem actually out working to treat patients today. Luke, you remind us why we do it. Winston, you and Tim have really galvanized the academic community. And, Chris, you and Creyon are really showing that, with time, we will be able to start creating engineering. We'll be able to create rules that bring efficiency. So I don't think any of us, in fairness, can say what the space will look like 5 years from now, 10 years from now. But with this kind of effort and this kind of mind, these kinds of motivations, we're going to see a lot of progress.
Thanks, Walt. Thank you.
Hey guys, I've met most of you, have not met you.
Yeah.
I started as a patient advocate for my daughter Rose, and thank you for sharing that, Luke, and being vulnerable. It means the world to all of us. Data is a very unemotional word, and so thank you for doing that. I have a little bit of an uncomfortable question, and it's primarily for nLorem and Creyon Bio. Because we look at incentives, we look at aligning incentives, we're talking about data sharing. We know with N-of-1s and just ultra-rare diseases. And with antisense technology, we know there's a lot that we don't know, especially when it comes down to toxicity. And now, I went from foundation, now running a lab for rare disease, foundations. And we all want to make the best drugs possible. Right? But my question to you, Sarah, and with Creyon Bio, you guys both have different incentives in terms of how much data you can share around the drugs you're developing. And I guess I would like to, I've heard Stan say nLorem or Ionis should be the only ones making ASOs because we know most about it. And I completely understand the history of you guys have more information and data than probably anyone in the world on this type of technology. But if we keep it to a small group of people and we don't teach others, then it's that 1,000-year approach, where it's going to be another 1,000 years before we get there. So could you talk about the type of data you're hoping you're going to be able to share and how that's going to look as a community so that we can expand our work?
Yeah, I'm happy to start, and I love uncomfortable, so thank you for bringing that up. I think the important thing here, just to be clear, when you say the toxicity is very unknown, there's actually a lot known. So that's the key point here, is the only chemistry that is actually enabled through these FDA guidance documents have many publications on the toxicity. If you make this chemical modification, if you change the sequence to this, all of these kind of nuances of antisense oligonucleotides and RNA therapeutics, most of it is published. And so I think that's one of the first elements, is just to be important. On cytotoxicity, a lot of that is published. And so I think that's where Stan's concern, of course, he pioneered and invented this 35 years ago, so has seen this grow and is so excited and inspired to, by the community to be really launching this. The concern is safety. It's quality and it's safety. And so there's no reason other than that to be able to try to ensure that we're all doing quality. I mean, from the drug discovery perspective, from the sharing and everything else, I think our goal is to, again, leverage the published data and to apply some of the tools that we've established internally and through a partnership with Ionis and Stan's work previously, as an independent organization to be able to most effectively and efficiently do this. And we do this in a very open fashion and in a nonprofit perspective. And so I think that's the thing. When you talk about sort of what are the end goals, what are the inspiration for our work, that's the reason why I love to work with nLorem, honestly, because I can stick my parent hat back on and say, you know what, truly and honestly, like our goal and is really to be open access, to be sharing our learnings, to publish often, to publish on each individual patient for the physicians to do that, to publish across ASOs, to collaborate with Winston and others. And I think really we don't have commercial goals. So I think there is clearly a difference. And our goal even isn't the drug approval. So, I can't comment on how that relates to having commercial goals, because that will never be. And nLorem really believes that at this point, at least, the nonprofit approach is the only way to be equitable for patients to be able to, if their mutation is amenable, to be able to help and have an ASO discovered and developed. But ultimately, over time, the goal is to be able to grow this, to share as much data as is needed, and in a place and in a platform and otherwise, I think through RareX and others, to be able to have the data open in a way that makes sense and is useful. And I think it's meaningful. And how is it, not just throwing data out there, but like in a publication, for example. I mean, I'd be happy to send the 4 publication database papers that are very specific, and they give you a lot of guidance on how to develop the safe ASOs.
I also think, just jumping on it as somebody who's involved very deeply with nLorem, is the education models that nLorem has put out there. I mean, there are these podcasts, there are, I have learned more than I did in 25 years of acting in conservatory in 5 years, and then in the last 6 months about science, in listening to the podcasts and listening to the educational resources that nLorem has put out there. So I would encourage people to learn more by checking out that website. It was really helpful for me to understand what is going into hopefully, my fingers crossed, daughter's body.
Yeah.
Were you gonna? Yes.
I'm happy to add a couple of words to what I don't think is an uncomfortable question. I think it's a good question. And I think we should take one step back and ask what are the data, what types of data would be important to share, what types of data are important for individual groups and individuals themselves, and then sort of how do we ensure that everyone is being we're facilitating the best process forward. And I think the thing that I will we're a commercial organization, obviously, with goals to making our platform capable of being efficient enough so that it is in fact a viable business, such that we can live within the healthcare system we have. Payers will pay for the drugs, and we can move forward. So that allows us to sort of have equitable access to everyone who could benefit from an oligo just by going to their doctor. It should be routine. That's our vision. And to do that, we obviously need to make the process of creating these medicines hyper efficient and to make them safe and efficacious. And we want to make sure we do that in a way that's beneficial to everyone involved. But the reality is today we can't do that. We're working to make that better. And one of the things that we challenge, which how do we be good citizens in the community to do this? And it comes from the realization that I think most people, if you've dabbled your foot into making oligos, you recognize that the concept is simple, right? You write a reverse complement, you get nice Watson-Crick-Franklin hybridizations to your RNA of choice, and magic should happen. It should be either knocked down or splicing should change or it should be modulated in some great way. And then you can read the literature and you can say, I got these chemical modifications that I can put onto them, and it will be well tolerated in the cell. The reality that we all know is that it matters where and what sequences you put in. You change a base, leave the chemistry the same. You can go from a perfectly safe drug to an incredibly toxic drug. You can do the same thing with chemistry. Take a sequence, changes chemistry a little bit. You go from a safe drug to a toxic drug or an active drug. So there's a lot of nuance associated with this, which is why we all don't have a capacity to make these drugs quickly. So just like Sarah was saying, I think that one of the most important things that we do as a community is that we ensure that if there are assays that we can develop that can better predict human safety. Right now, the way that drugs are developed is we do in vitro work, we do in vivo work and non-clinical studies, and then we go into patients. And the sort of war, not just for oligos, but for all drugs, is that sometimes that process that we went through didn't work, and we end up with a toxicity in humans. And the last thing anybody, I think, wants is that to happen. So if there's a way that we can measure something about a compound that gets created before it goes into a human that would be indicative of being safe, or not safe, and that can be shared. That should be shared immediately, and it should be shared openly. And I think the community actually does a pretty good job with the science, and there's a lot of publications, and I think that's one of the places where we can do that. And if there's other forums that can be brought into play to make sure that there's the right control compounds, the right ways of evaluating oligos that level the playing field and make sure any compound that's made has the best chance of being safe. We should be always pushing forward on that. And then the second aspect of how do you keep, how do you ensure though that organizations have, their other objectives are being met? I think one of the things that we should keep in mind is that we all walk around in a commercial world where we're benefited by the sort of the markets that exist, right? But every one of us has a pocket computer in their phone, in their pockets, which is running on silicon chips. It's not that people didn't know about silicon, the idea of what a transistor was, how to build these transistors, and the sort of physical properties of the transistors. Those are all published in scientific literatures and exchanged freely. What started to emerge was how do you assemble those into circuits that have meaningful processing power? How do you turn those into things that we can make really fast? And there's proprietary information that lives in that know-how, and there's patents that get filed, there's trade secrets get retained, and allows companies to have profit, which allows for efficiency so that you can get these things out to everybody in a way that's affordable and distributable within the systems we live within. So I think there's a balance here that we need to reach, which is sharing things, and we should be always sharing things to ensure that we're helping people be safe and be the best they can be. But we also want to make sure we don't encumber businesses to do what businesses can do very efficiently, which is make things cheaper and faster so they can be distributed and equitably distributed.
Thank you, and I'm sure we need to get going.
Final word?
Well, I was just going to close it out because we should get going, right? No, I was thinking back to you, right back to you.
Okay, well, thank you. Extraordinary, passionate, thinking outside the box, the future. You guys have it all, and Luke, we love you so much. Thank you for, like Casey said, always being vulnerable. Thank you to you all. Let's please give them a round of applause. We are running a little bit behind, but we're going to make it up. While we bring up Charlene and Indu, I would love for everyone to stand up and stretch or something, do a little jog. Where's my Graylia? Do we have any did he step out? And of course he did, right? Do a little yawn, a little stretch. Yeah. And if you need to hop for a bio